Our next speaker is Dr. Heath Schmidt, an assistant professor in the Department of Biobehavioral Health Studies in the School of Nursing and the Department of Psychiatry in the School of Medicine. He received his PhD in pharmacology and neuroscience from Boston University School of Medicine, studying the role of dopamine, glutamate, and acetylcholine in animal models of cocaine addiction. Dr. Schmidt completed his postdoctoral training at Yale University in the lab of Dr. Ron Duman, studying the behavioral and cellular mechanisms underlying depression and anxiety. Dr. Schmidt has been awarded numerous grants from the National Institute on Drug Abuse, as well as from the University of Pennsylvania. His lab currently focuses on identifying novel neurobiological mechanisms underlying nicotine, cocaine, and prescription opioid addiction-like behavior. The question he'll be asking tonight is, can anti-diabetic drugs be used to treat cocaine addiction? Please welcome Dr. Heath Schmidt.
Can you turn the lights off for my two IHC slides? All right, can everybody hear me? How about up in the nosebleed seats? We good? All right, awesome. All right, so thank you, Amanda, for the introduction. Thank you, NGG, for organizing tonight's public lecture, and thank you all for coming out tonight. Mariella gave a great talk on a drug of abuse. We're going to switch gears and talk about an illicit drug of abuse, cocaine, and the potential for anti-diabetic drugs to treat cocaine addiction. Now, cocaine is the third most commonly abused illicit drug in this country, only after marijuana and prescription opioids. The U.S. is the world's leading consumer of cocaine. We buy about $28 billion of the drug annually. Okay? And currently there are 2 million regular cocaine users in the U.S. This number is increasing. So the map that you see here shows the percentages of young adults aged 18 to 25 years reporting having used cocaine in the previous year. Now the average is 5% of this population in the country, and that represents a 64% increase in young adults that have used cocaine compared to the same age group in 2013. And it's a 24% increase compared to the same age group in 2014. Okay? And it's not just specific to this demographic. Cocaine use is also up in females of all ages, as well as the baby boomer generation. And it's not entirely clear why. Some have speculated because it's due to the opioid epidemic. So some individuals, this is misguided, but view cocaine as being a safer alternative to opioids. But that's not clear. Now, unfortunately, there are no FDA-approved medications to treat cocaine addiction. Right? And that's one mission of our lab, is to try and identify new drugs that could be used to treat cocaine addiction. Now, cocaine addiction is a chronic relapsing disorder consisting of 3 phases. The first is a period of chronic or compulsive drug taking, and that's followed by voluntary or involuntary detoxification. And it's during this withdrawal or abstinent period that an addict will experience an intense psychological craving for the drug. And this craving can promote relapse back to chronic drug taking. Now, relapse is the universal hallmark of cocaine addiction. So if we were all clinicians and we were treating cocaine addicts, this is the point that we would try to intervene. We would try to prevent or reduce relapse rates in our addicts. Now, relapse is a significant problem, as I said. 80% of cocaine addicts will relapse to chronic drug taking within the first 3 months of successful detoxification. And there's 3 stimuli that can promote relapse in human cocaine addicts. The first are drug-associated cues. So these are aspects of the environment that, when paired with cocaine taking, become rewarding in their own right. So this could be the environment in which cocaine was taken. It could be drug paraphernalia, or it could be your friends that you use cocaine with. Okay? So re-exposure to all of these things during withdrawal can cause an addict to crave the drug and could promote relapse back to chronic drug taking. Now, the second stimuli that could precipitate relapse is a stressful life event. So perhaps your friends abandon you and they no longer defend your bad habits. Okay? That could promote relapse as well. And finally, re-exposure to the drug itself. So our lab is very much interested in understanding ways to intervene here at this relapse stage. Now, the way that we model relapse and the way that we study the brain and the mechanisms in the brain, okay, that might mediate craving-induced relapse is by using an animal model, and it's called the reinstatement model. Now, the reinstatement model relies on drug self-administration in rodents. So our favorite subject to study is the laboratory rat. Now, we anesthetize our rats, and we surgically implant them with a catheter such that one end of the catheter resides in the animal's heart, and the other end of the catheter exits through a port on the animal's back here. Now, following a recovery period, we can put our rats into these operant chambers, like the one you see here. We attach the catheter to a series of tubing that lead to a syringe that contains a cocaine solution. And this syringe is housed in an automated pump. Now, at this point, we shut the door and we walk away, and we just leave the animal alone. Now, rats are normally curious creatures. So he's going to explore this chamber, and at some point he's going to bump into and press a lever that's protruding into the chamber. And this photograph captures one of our rats pressing this lever. When he presses this lever, the pump fires, it depresses the plunger on the syringe, and thereby giving the rat an IV infusion of cocaine. Okay? So we're not injecting our animals with cocaine, and that's an important distinction with this model compared to other models that are out there. You can think of us as the drug dealers. We supply the cocaine, but we don't actually give it to the animal. They're in charge of their own pattern of cocaine use. So this is how we model voluntary cocaine taking in rats. Now, why is a rat going to press a lever for an infusion of cocaine? For the same reason a human is going to take cocaine, because it makes him feel good. All right, so self-administration models voluntary drug taking in rats. So if we look at a group of rats that are self-administering cocaine for 3 weeks, what you can see is that toward the end of this 3-week period, our rats are pressing the lever on average about 140 times. So this is equivalent to about 28 infusions of cocaine. Okay? So our rats love their cocaine, they like to press the lever when it's available, and they get a lot of it. Now at this point we can put our animals through our version of rodent rehab. So this is a withdrawal period in which we take cocaine out of the syringe and we replace it with saline. So now when the animal goes into the box and presses the lever, he gets an infusion of saline. Well, saline doesn't feel like anything. It certainly doesn't feel like cocaine. So he's going to stop pressing the lever, and that's what you find. Now, after 7 days of withdrawal, we can reinstate drug seeking in our rats, and we can do that by re-exposing them to cocaine. Remember, this is one of the stimuli that promote relapse in human cocaine addicts. So what we do is we give our animals, our rats, an acute injection of cocaine, and then we put them into the operant chamber. And what we find is now our animals press the lever like crazy. Okay? Bear in mind they're only getting saline on this day. So what's the difference? The difference is they got this little hit of cocaine to start. All right? So if I was to anthropomorphize what's going on here, that little hit of cocaine is causing our rats to crave the drug. So when they go into the operant chamber, they remember, hey, this lever used to give me cocaine. So now I'm going to press it. And they press it like gangbusters, almost to the same levels that you see when cocaine's available during the self-administration phase. Okay? And they're doing that because they want the drug. They're craving the drug. So they're pressing the lever, trying to find that drug again. So this is how we model relapse or drug seeking in our animals. Now, cocaine addiction is a disease of the brain. Cocaine gets into the brain and it increases dopamine signaling. And it does this by activating the reward circuit in the brain. So the reward circuit consists of dopamine neurons in the VTA that project to the nucleus accumbens. Now this reward circuit is the same circuit that mediates the rewarding effects of natural rewards, like food, sex, and social interactions, all the things required for our species to survive. The difference is that when cocaine gets in there and hijacks that reward circuit, it boosts dopamine signaling to levels much higher than what's seen following intake of food or during sex or when you're having a social interaction. Okay? So we're very much interested in understanding mechanisms that might dampen down this reward circuit as a way to try and reduce cocaine seeking in our rats, okay, and hopefully relapse in human cocaine addicts. Okay. So we became interested in this hormone called glucagon-like peptide 1. It's a satiation hormone. It's released by the small intestine. So when you eat a meal, the meal travels down to your GI tract and GLP-1 is released. And it has a number of functions including lowering blood glucose levels and reducing food intake. So it's not surprising to learn then, and some of you may have experience with this, that GLP-1 analogs like Byetta and Saxenda are FDA approved to treat type 2 diabetes. And in Saxenda's case, it's also approved for weight loss in morbidly obese individuals. Now, GLP-1 receptors are also expressed in that reward circuit that we were talking about. And GLP-1 can get into the brain and activate those receptors. And then finally, what I want to point out is that there is a source of GLP-1 in the brain, and it's in an area of the brain called the nucleus tractus solitarius, or the NTS, and we'll come back to that. Okay. So recent data suggest that if you activate GLP-1 receptors in the reward circuit, you can reduce intake of highly palatable food. So why that's interesting to us as addiction scientists is for the same reason that cocaine hijacks the same circuitry in the brain. So this suggests that if we take a GLP-1 receptor agonist, inject our animals, it should get into the brain, bind to receptors in the reward circuit, and dampen down or decrease cocaine seeking in our rats. Okay? And that was exactly what our hypothesis was going into the study. So administration of a GLP-1 receptor agonist would reduce cocaine seeking in the rats. And the design was just like the model that I described. We had animals self-administer cocaine for 21 days. We then put them through our withdrawal period. And then we treated them with Byetta, also known as Exendin 4. And we did this prior to that small hit of cocaine. And what we were predicting is that Byetta would decrease this blue bar here, indicating a reduction in cocaine seeking in our rats. And that would be analogous to reduced relapse in human cocaine addicts. Okay? So that's the experiment that we did. And I want you to focus only on data under this active lever label here. So this white bar reflects animals that are treated with placebo and then given their primary injection of cocaine. And you can see that rats are responding about 80 times on the lever. So that's robust cocaine-seeking behavior. They're pressing the lever because they're looking for the drug. Now, if we pre-treat the animals with Exendin 4, clearly you see this reduction in cocaine-seeking behavior. And this was really exciting to us because this is the first demonstration in an animal model that these anti-diabetic drugs could potentially be used to treat an addiction-like behavior, specifically reduce craving-induced relapse. Now, we had to temper our enthusiasm a little bit because these drugs are commonly known to produce side effects in humans, most notably nausea. They also reduce body weight and food intake. So we wanted to be able to screen our animals to see if these same side effects were occurring at the doses that we were giving. So we measured body weight and food intake, and we found no effects at the doses that blocked cocaine seeking. So this really got us excited because it suggested a new use for these drugs, and it seemed to indicate that they could be selective for blocking cocaine seeking and not have these adverse effects that are known to be associated with these drugs in humans. Now, as I said before, cocaine addiction is a disease of the brain, and GLP-1 receptors are expressed in the brain. So we wanted to know whether or not our agonist was getting into the brain, activating GLP-1 receptors, and producing that behavioral response. So we dissected the brains of our rats, and we looked in the nucleus accumbens and the VTA. And hopefully you can see this pretty clearly. So what you're looking at in these two photographs here, thank you, whoever's doing that, So what you can see in these two photographs are a bunch of green dots. Each green dot represents a molecule of exendin-4, our GLP-1 receptor agonist. These blue blobs are the neurons in the nucleus accumbens in the VTA. So clearly our drug is getting into the brain and it's binding to receptors expressed on the VTA and the nucleus accumbens. Now this is just a different, a more magnified image that I'll show you where you can see three-dimensionally this binding to the neurons. Okay? So this was encouraging because it's a clear demonstration that our drug is getting into the brain, but it doesn't really provide direct evidence that activating these receptors in the reward circuit could reduce cocaine seeking. So we devised another experiment to look at this a little more directly. So instead of giving the animals a systemic infusion of exendin-4, what we did was during withdrawal, we actually infused the drug directly into the brains of our rats. And we did so in both the nucleus accumbens and the VTA. So the idea being that we wanted to see if activating these GLP-1 receptors in these two brain regions in the reward circuit was sufficient to reduce cocaine seeking. And that's in fact what we found. Okay? So I'm going to describe one more experiment to you, and that relies on the endogenous GLP-1 circuit within the brain. So remember I told you that there was a small population of neurons in the NTS that express GLP-1? Well, it turns out that these neurons project to the reward circuit, the VTA, and they release GLP-1 here. So I just showed you evidence that increasing GLP-1 signaling in the VTA should reduce cocaine seeking. So we began to wonder if we could use a gene therapy approach to try and activate this endogenous circuit in the brain. So avoid the drug altogether and instead try to manipulate this circuit so that we could increase GLP-1 signaling in the brain. And the way that we did that was with a new technology called DREADS. So DREADS are receptors that are not expressed in any tissue in our body. This DREAD, when it's expressed by a cell, will activate that cell. We decided to package this DREAD into a virus, okay? With the idea being that if we infuse the virus into the brain, specifically into the NTS, the virus will bind to these neurons, inject the DREAD receptor, and then we'll see neurons in the NTS now expressing this receptor. Okay? And just to show you a little bit of evidence for that, here's an image, and all these green cells that you see fluorescing are neurons that are expressing that DREAD protein. All right? So now what can we do with this? Well, what we can do is we can inject our rats during withdrawal with a compound that normally has no effect on its own, but when it binds to these DREAD receptors, will activate these cells. So we should get increased GLP-1 release in the reward circuit. Okay? And that's what we did. And what we found was that indeed, if we're able to activate this circuit in the brain, we can reduce cocaine-seeking behavior in our rats. Okay? So to summarize, hopefully I've convinced you at this point that we've identified a new use for a GLP-1 receptor agonist. We've identified doses of an agonist that selectively reduce cocaine-seeking behavior in rats that are not associated with adverse effects that are commonly seen in humans. I've also showed you evidence that increased GLP-1 signaling in the brain may represent a brake that functions to reduce cocaine craving-induced relapse. So what we think is going on is that when we treat animals with Byetta, during withdrawal, it's getting into the brain, it's increasing GLP-1 signaling in this reward circuit that's decreasing the reward circuit, and ultimately that's reducing cocaine seeking in rats and hopefully relapse in human cocaine addicts. So we're excited as basic scientists because I think these data support clinical trials of GLP-1 receptor agonists in cocaine addicts. And we've actually initiated a small pilot trial of these compounds in human cocaine addicts. Now, I don't have any data to show you because we just started this. So instead, I'm going to leave you with that as a cliffhanger. Right? So maybe I can come back next year and share that with you. All right, so I want to acknowledge the people whose hard work allowed me to stand up here and talk to you tonight. Kelsey Ige was an undergrad in the lab that helped me pilot these experiments. Nicole Hernandez took the reins, and she's really responsible for the really nice story that's coming out of this project. Nicole is a fourth-year neuroscience graduate student here at Penn. Nicole's had an assist from Giancarlo Molina, who is a summer intern from Puerto Rico, and a current undergrad in the lab, Vanessa Weir. I'd like to thank my funding sources, NIDA, as well as CURF, and thank you all for coming out tonight.
Thank you, Dr. Schmidt. We have time for 2 questions this time.
To what extent is the lever pressing an indication of cocaine seeking versus an overall increase in motivation and motivation to just press things and explore?
So that's a great question. So the question is, to what extent is lever pressing an indication of cocaine seeking versus motivation for the drug? So there are different ways to look at motivation in cocaine seeking. I would say that the model that I described to you certainly incorporates both. It's definitely a model of drug seeking as I've described, but obviously the animal has to be motivated to some degree to seek that drug. So the precipitating stimulus for that is that acute hit of cocaine at the beginning, right? So there are other ways to study motivation as well. There are other models. I haven't described them. They're somewhat complicated, but we can assess motivation directly using some other models in the lab. Okay. Yep.
In your plots of lever pressing for over days, there was like a really discrete jump between day 5 and 6. Is there a physiological mechanism for that?
No, so that's just that's methodological. So I didn't want to get into the details, but on days 1 through 5, the animal's only required to hit the lever 1 time for every infusion. Then we increase the complexity of the task. So at day 6, they have to press the lever 5 times for every infusion. So basically they're getting the same number of infusions on that first week, and in weeks 2 and 3, they just have to hit the lever 5 times more to get it. And that just provides us with greater spread in the data so that it's more sensitive to manipulations. Yeah, good question.
Are you aware of any less invasive mechanisms to drive GLP-1 expression?
Well, the agonists, certainly. I mean, just targeting the receptors themselves, so there's the pharmacological approach to doing it. Other than that, no. I mean, there's transgenic models in mice that you could use, but no. Mm-mm.
What does the BTA project to? That is, actually in circuit, what term of the larger system it activates?
What does what project to? I didn't hear.
VTA,
The VTA?
Yeah.
So the VTA sends dopaminergic projections to the nucleus accumbens, so that's part of the reward circuit. But it also projects to other places like the hippocampus, okay, and dopamine signaling there is involved with the memories associated with drug taking. It also projects to the prefrontal cortex, which is responsible for impulsivity, right? So you can imagine all of these things working together to drive addiction-like behaviors. Okay?
That's all the time we have. Thank you so much, Dr. Schmidt.